Good afternoon and welcome back to AP Biology, Semester 2, and we're on Big Idea 2, Free Energy. Today we are in Chapter 7, Section 3, talking about the passive transport of diffusion being the substance across a membrane with no energy investment. Okay. Molecules have a type of energy called thermal motion, or heat. One result of thermal motion is diffusion, the movement of molecules of any substance so that they spread out evenly into the available space. Each molecule moves randomly, yet diffusion of a population of molecules may be directional. To understand this process, let's imagine a synthetic membrane separating pure water from a solution of a dye in water. Assume that this membrane has microscopic pores and is permeable to the dye molecules. Each dye molecule wanders randomly, and there will be a net movement of the dye molecules across the membrane to the side that began as pure water. The dye molecules will continue to spread across the membrane until both solutions have equal concentrations of the dye. Once that point is reached, there will be a dynamic equilibrium with as many dye molecules crossing the membrane each second in one direction as in the other. We can now state a simple rule of diffusion. In the absence of other forces, a substance will diffuse from where it is more concentrated to where it is less concentrated. Put another way, any substance will diffuse down its concentration gradient, the region along which the density of a chemical substance decreases. No work must be done in order to make this happen. Diffusion is a spontaneous process, needing no input of energy. Note that each substance diffuses down its own concentration gradient, unaffected by the concentration differences of other substances. The diffusion of a substance across a biological membrane is called passive transport because the cell does not have to expend energy to make it happen. The concentration gradient itself represents potential energy and drives diffusion. Remember, however, that membranes are selectively permeable and therefore have different effects on the rates of diffusion of various molecules. In the case of water, aquaporins allow water to diffuse very rapidly across the membranes of certain cells. As we'll see next, the movement of water across the plasma membrane has important consequences for cells. Okay, so going on, we are going to talk about the effects of osmosis on water balance. To see how two solutions with different solute concentrations interact, picture a U-shaped glass tube with a selectively permeable membrane separating two sugar solutions. Pores in this synthetic membrane are too small for sugar molecules to pass through, but large enough for water molecules. How does this affect the water concentration? I'm so glad you asked. It seems logical that the solution with the higher concentration of solute would have the lower concentration of water, and that water would diffuse into it from the other side for that reason. However, for a dilute solution, like most biological fluids, solutes do not affect the water concentration significantly. Instead, tight clustering of water molecules around the hydrophilic solute molecules make some of the water unavailable to cross the membrane. It is the difference in free water concentration that is important. In the end, the effect is the same. Water diffuses across the membrane from the region of lower solute concentration to that of higher solute concentration until the solute concentrations on both sides of the membrane are equal. The diffusion of water across a selectively permeable membrane is called osmosis. The movement of water across cell membranes and the balance of water between the cell and its environment are crucial to organisms. Let's now apply to living cells what we have learned about osmosis in artificial systems. Okay? We're going to talk about water balance of cells without walls. Okay? But I want to reiterate before I go on that water moves from an area of higher to lower free water concentration, lower to higher solute concentration. Okay? Just wanted to reiterate that. Okay? So, when considering the behavior of a cell in a solution, both solute concentration and membrane permeability must be considered. Both factors are taken into account in the concept of tonicity, the ability of a solid solution to cause a cell to gain or lose water. The tonicity of a solution depends in part on its concentration of solutes that cannot cross the membrane, non-penetrating solutes relative to that inside the cell. If there is a higher concentration of non-penetrating solutes in the surrounding solution, water will tend to leave the cell and vice versa. If a cell without a wall, such as an animal cell, is immersed in an environment that is isotonic to the cell, iso means same, there will be no net movement of water across the plasma membrane. Water flows across the membrane, but at the same rate in both directions. In an isotonic environment, the volume of an animal cell is stable. Now let's transfer the cell to a solution that is hypertonic to the cell. Hyper means more, in this case referring to non-penetrating solutes. The cell will lose water to its environment, shrivel, and probably die. This is one way an increase in the salinity or saltiness of a lake can kill animals there. If the lake water becomes hypertonic to the animal cells, the cells might shrivel and die. However, taking up too much water can be just as hazardous to an animal cell as losing water. If we place the cell in a solution that is hypotonic to the cell, hypo meaning less, water will enter the cell faster than it leaves, and the cell will swell and lyse or burst like an overfilled water balloon. A cell without rigid walls can tolerate neither excessive uptake nor excessive loss of water. This problem of water balance is automatically solved if such a cell lives in isotonic surroundings. Seawater is isotonic to many marine invertebrates. The cells of most terrestrial land-dwelling animals are bathed in an extracellular fluid that is isotonic to the cells. Animals and other organisms with rigid cell walls living in hypertonic or hypotonic environments must have special adaptations for osmoregulation, the control of water balance. For example, the protist, Paramecium, lives in pond water, which is hypotonic to the cell. Paramecium has a plasma membrane that is much less permeable to water than the membranes of most other cells. But this only slows the uptake of water, which continually enters the cell. The paramecium cell doesn't burst because it is also equipped with a contractile vacuole, an organelle that functions as a bilge pump to force water out of the cell as fast as it enters by osmosis. We will examine other evolutionary adaptations for osmoregulation in chapter 44. All right, so we're going to go on to the water balance of cells with walls. The cells of plants, prokaryotes, fungi, and some protists have walls. When such a cell is immersed in a hypotonic solution, bathed in rainwater, for example, the wall helps maintain the cell's water balance. Consider a plant cell. Like an animal cell, the plant cell swells as water enters by osmosis. However, the relatively An elastic wall will expand only so much before it exerts a back pressure on the cell that opposes further water uptake. At this point, the cell is turgid, very firm, which is the healthy state for most plant cells. Plants that are not woody, such as most houseplants, depend on mechanical support on cells kept turgid by a surrounding hypotonic solution. If a plant cells and their surroundings are isotonic, there is no net tendency for water to enter and the cells become flaccid or limp. However, a wall is of no advantage if the cell is immersed in a hypertonic environment. In this case, a plant cell, like an animal cell, will lose water to its surroundings and shrink. As the plant cell shrivels, its plasma membrane pulls away from the wall. This phenomenon, called plasmolysis, causes the plant to wilt and can lead to plant death. The walled cells of bacteria and fungi are plasmolyzed and in hypertonic environments. Alrighty, so now we're going to move on to facilitated diffusion, passive transport aided by proteins. And then we're going to stop for today. Okay, so let's look more closely at how water and certain hydrophilic solutes cross a membrane, how they cross a membrane. As mentioned earlier, many polar molecules and ions impeded by the lipid bilayer of the membrane diffuse passively with the help of transport proteins that span the membrane. This phenomenon is called facilitated diffusion. Cell biologists are still trying to learn exactly how various transport proteins facilitate diffusion. Most transport proteins are very specific. They transport some substances, but not others. As described earlier, the two types of transport proteins are channel proteins and carrier proteins. The channel proteins simply provide corridors that allow a specific molecule or ion to cross the membrane. The hydrophilic passageways provided by these proteins can allow water molecules or small ions to flow very quickly from one side to the membrane to the other. Although water molecules are small enough to cross through the phospholipid bilayer, the rate of water movement by this route is relatively slow because of the polarity of the water molecules. Aquaporins, the water of channel proteins, facilitate the massive amounts of diffusion that occur in plant cells and in animal cells such as red blood cells. Kidney cells also have a high number of aquaporins, allowing them to reclaim water from urine before it is excreted. It has been estimated that a person would have to drink 50 gallons of water a day to excrete the same volume if the kidneys did not perform this function. Another group of channel proteins are ion channels, many of which function as gated channels, which open or close in response to a stimulus. The stimulus may be electrical or chemical. If chemical, the stimulus is a substance other than the one to be transported. For example, stimulation of a nerve cell by certain neurotransmitter molecules opens gated channels that allow sodium ions into the cell. Later, an electrical stimulus activates the ion channel protein, and potassium ions rush out of the cell. Carrier proteins, such as the glucose transporter mentioned earlier, seem to undergo a subtle change in shape that somehow translocates the solute binding site across the membrane. These changes in shape may be triggered by the binding and release of the transported molecule. In certain inherited diseases, specific transport systems are either defective or missing altogether. An example is cystinuria, a human disease characterized by the absence of a carrier protein that transports cysteine and some other amino acids across the membrane of kidney cells. Kidney cells normally reabsorb these amino acids from the urine and return them to the blood. But an individual afflicted with cystinuria develops painful stones from amino acids that accumulate and crystallize in the kidneys. Otherwise, or known as kidney stones. And we are going to stop here for today. The next time we meet, we will be going to section 7.4, talking about active transport using energy to move solutes against their gradients. Okay, and until next time, ta-ta!